Yeah. Okay. I'm sure we're going to get a bunch of people walking in like in 10 minutes because that's what always happens with these things. But thanks very much, Andy, for coming and giving the ERI seminar. Are we recording this as well?
Yeah, it's
yep.
already started.
Yep.
I will turn up
the banter at the beginning.
Yeah.
Lovely. Thanks. So yeah, that means that people can check it out later on as well if they haven't, for teaching would have been able to come in person. So Andy's Professor of Biosciences at University of Cardiff. I got to know Andy through the Cardiff Waikato what's called Strategic Initiative, and he's been part of the XNA project a little bit peripherally with giving some sequences from the deep sea biosphere project, and so been able to look for add-ins onto that. But today he's going to be talking about his work with the Welsh Government on wastewater testing for COVID-19, and I think this is just a fantastic title.
I'll come on to that, yeah.
Yeah, thanks very much.
Okay, well, thanks for the invitation. It's lovely to be here. I'm not sure. I mean, in some ways I'm here, and in some ways I'm mid-Pacific. I don't know where I am, to be honest. But I arrived yesterday, sun shining. You know, what's not to like? So thank you very much. And what I'll be, as Adele said, what I'll be talking about today is really just what we've called the WEWASH Programme, which it's a shortened form of Wales Wastewater Sentinels for Health and Environmental Surveillance, but really started under COVID, as I'll explain. And just to illustrate, I hope, some of the benefits and the challenges of working with government, and in particular the Welsh Government. How we've progressed that, and where we are at the moment, indeed. So there is a wastewater programme, and has been for some time in New Zealand. I can't remember the initials of the organisation that runs that. It's ESR or something. Yeah. And they're still running. I saw that the report for January. We're in a bit of limbo at the moment. We're just running towards the end of March, and we're not quite sure what's happening politically, but more about that anon. So, there we are. So it'll be a lot about wastewater, poo, and a tiny bit about how that's sort of translated in terms of policy. And pandemic preparedness is where the whole world should be at the moment, not necessarily for another virus pandemic, but AMR is in the background, always has been, and these sorts of things, need some sort of serious consideration, obviously. So the original project started through a conversation that I had with a colleague at the University of Bangor, Davy Jones, who's a soil microbiologist and has been working on environmental virology through a postdoctoral fellow called Kata Farkas, and liaising with various colleagues in the water utilities, Welsh Water in particular. And this map just shows the area that's covered by Welsh Water, and the blobs are the wastewater treatment works, large, medium and small, across the whole of Wales. The main water treatment works are in Cardiff, and for the south of Wales, and Wrexham for the north of Wales. But as you can see, there are hundreds of the things, and the decision that we had to make early on was how many to look at. It also involved funding from Welsh Government, so relatively quickly, and this was great, they responded. In England, there was a project that was going on at the same time, but probably not as well coordinated in terms of links between government and public health. So we involved a Public Health Wales, and Dŵr Cymru Welsh Water is the main water company for Wales, but there's also a Severn Trent outlet that controls this area of Wales. As you probably know, or as you might know, these days wastewater can be used to measure all the activities of society, really. We can look at what we're producing in terms of waste, chemical, pharmaceuticals, antibiotics, You can measure products from that have come in through being thrown into the wastewater, or just derivatives of human activity in some shape or form, endocrine disruptors, hormones, and so on, and pathogens, pathogenic bacteria, pathogenic viruses as well. And wastewater-based epidemiology, therefore, had been is a concept and had been applied quite widely. But the question was, and it had been used for in various ways, and to look at poliovirus in particular. I'll mention that in a moment. Asymptomatic and pre-symptomatic, and data existed for some viruses, particularly norovirus. So in Bangor, they were doing quite a lot of work looking at norovirus in the wastewater. But at the outset, it wasn't really known whether you could measure SARS-CoV-2 virus, the COVID virus, in wastewater. So would this work for SARS-CoV-2? Not necessarily, obviously, good to look at in wastewater because it's a respiratory virus. You know, to what extent would it be transmitted through wastewater? Would it come through urine? Would it come through feces? It turned out, of course, it was quite easy to detect it in fecal matter and therefore in wastewater. So surveillance of using wastewater were developed in the 1940s, so way back. Cell culture methods were used to isolate poliovirus from wastewater, and that was really, and I suppose a bit of work with hepatitis A in the US in the 1980s, but things really changed with the advent of the polymerase chain reaction, PCR, where we could start to design primers for all sorts of different agents in wastewater, and particularly taking up the work with hepatitis A and norovirus and polio through the 2000s. But the thing that's given it real impetus, and I think hopefully will be good for the future, a legacy, as it were, of something that was really terrible, is as a legacy of the COVID-19 pandemic. And this shows some early work. It actually was published, but quite late in, let me see now, in 2020, of course. But this was done by the MEDMA group in the Netherlands. And I remember seeing a presentation, it was a town hall, so-called town hall presentation, EU town hall presentation online. And Gertjan was talking about the work that they'd done around three wastewater treatment works around Utrecht and Amsterdam in the Netherlands. And what this figure shows is the number of reported cases that were occurring right then, back in the pandemic. I mean, before lockdown in the UK, and I can't remember when lockdown happened in New Zealand, but relatively early stages, late February, early March, and you can see the cases increasing. Scale on the left-hand side here, and then the virus detection, SARS-CoV-2 virus detection, which was fairly rudimentary at that stage in wastewater treatment works around Utrecht and Amsterdam, showing the gene copies per mil of wastewater on the log scale on the right-hand side. So this was really gave us the confidence to invest quite a bit of effort and governments to put quite a bit of money into detecting the virus in wastewater in other countries. So this is a summary of how the programme has, as it were, developed and where we are now through about 5 phases of the programme, starting although we were talking to Welsh Government back in March, April of 2022, but the funding really started to come on stream in July for a pilot project, and then through these phases to where we are now, and I'll say a bit about that at the end. But the total funding is something that, for the whole programme, and the partners that are involved are shown on the right-hand side, is really so substantial. I don't think I've ever had funding like that that's been given so quickly for any sort of project. And that was split mainly between Bangor and Cardiff over the period. So that's definitely an upside. The challenges are really, how do you get the information in a usable form to Welsh Government and to public health officials? And what government tends to do is to set up these huge organizations, or rather project management. And so we suddenly became a supplier to the user groups. And I suppose that it's particularly in the early stages, a problem was that as suppliers, we were supplying data and information to users who were the public health scientists and the public health officials. And Welsh Government was managing that relationship. And we weren't really talking directly, at first anyway, to the people involved. It also involved technical advisory group, which was the kind of providing scientific advice to Welsh Government that Davey and I both sit on, and test, trace and protect, which was basically doing clinical analysis in the community, and we had to interact with Chief Medical Officer and the Chief Scientific Officer for Health, who's Rob Orford. Really great people who put a lot of time and effort into the project and allowing us to move at pace, because there was a lot of innovation required as well. And I know a lot of countries were doing this work, not least New Zealand, European countries across the states, but it was difficult to coordinate it at the time because of the pandemic. And we were lucky, we had quite a bit of publicity that helped us raise the profile of the programme on the BBC and locally. And the benefits of doing this, I think, were fairly evident from the beginning. It was going to be and is, and we can show this now, much more cost effective than doing surveillance by testing. Catches asymptomatic as well as symptomatic and pre-symptomatic individuals. Automation, as we introduced it, robotics, for example, allowed us to process large numbers of samples relatively quickly, non-discriminatory. So these factors, I think, are particularly important by comparison with the problems we faced with testing individuals by PCR and the lateral flow test and getting those data back from all sectors of society. That wastewater, everyone has to poo, wastewater is non-discriminatory, independent of health-seeking behaviours, and independent of clinical testing. And that's really important, particularly when you're looking at some sectors of society, some areas geographical within Wales in particular. And the question as well at the beginning, although I don't think this is, I think the evidence lies rather on both sides. Were we able to use it as an early detection mechanism? That was certainly hoped at the beginning. And I think to some extent it's true that wastewater does provide an early indication of the signal coming through, and you can customize it as well. So we started looking at a bunch of sites as far as we could. Travel in those days was difficult because of the COVID restrictions. And we had to get drivers to move out to these sites. It involves collaboration with Welsh Water to go to the wastewater treatment works. But basically along a road here, which is the M4, for those of you who know Wales at all. And then there's another major road that goes along the top here. So that's what the drivers were doing, going up and down this every day, collecting samples from these 27 sites. And we quickly managed to expand with Welsh Water's help to about 50 sites, or just under 50 sites across the whole of Wales. In the middle of Wales here, there are dragons, so it's rather it takes you ages to get there, and it's well, there are sheep actually, to be honest. But all of these sites were being monitored 5 days a week, and we had the flexibility to taking samples from automated samples. We were using what's called composite sampling, so the sample was taken across the 24-hour cycle. You weren't just going and taking a spot sample at one particular time. So it was integrated signal over the 24 hours, 5 days a week, and weekends when necessary. And believe you me, it was necessary at sometimes to go at weekends. The thing that we didn't manage to push, and we would have liked to have done, was to be doing more sample within the networks, within the larger networks in particular. And I'll say something about this in a moment. But we did develop approaches for monitoring other sources, seaports, for example, at Holyhead and Fishguard. Airports, there's only one airport in Wales, which is Cardiff, but we're looking at Bristol Airport as well. And ultimately about 9 or 10 hospitals that were dotted around the major health boards of Wales. There are 7 health boards in the area. And we were breaking this down. So this map shows where the sites are. And there are 14 areas here that are coloured. And these aren't health boards, they're not political areas, they're actually the catchments for the main rivers in Wales. And it made more sense to, in fact, Welsh Government wanted us to focus on a geographical catchment rather than link it politically to certain areas of around the country for good reasons. So these are what are called the L2 drainage areas that are being identified by the water companies. So this infographic really was used to, when we started reporting, was developed to indicate what we were doing, collecting samples from all around Wales, pooling the samples in Cardiff and Bangor, and then carrying out the analysis, which initially was qPCR analysis of the SARS-CoV-2 virus in the lab here. And you can see these are the robots that we used for the processing of the samples, MSC microbial safety cabinets in which the work was done. No people there at the moment, but believe there were people there. And then I won't run through the protocol in detail, but I'm happy to answer any questions. But basically, you take your wastewater sample coming in. The most efficient way to identify the virus was to precipitate the RNA with polyethylene glycol. That involved leaving it overnight and then spinning it down. And that then could go onto an automated RNA extraction, DNA and RNA extraction, and then RT-qPCR to measure the virus signal, and retaining the sample as well, ultimately, once we've developed the sequencing procedures. So we're sampling these 47 sites, 5 days a week, and we could process samples collected on a Monday and be reporting the data by Wednesday, which was about 50 hours from collection to providing the detail for Welsh Government. And they were using it for policy as well, as it turned out. We didn't know it for a long time because they didn't feed back. But it's that there's a COVID-19 inquiry going on in the UK at the moment, which will ultimately report in 2026. It's really extensive. A lot of the publicity that you might have come across from the UK about that has been what's in the WhatsApps, you know, and people slagging off politicians, I should say, slagging off each other. But there is a serious purpose to this inquiry, and the ministers in Wales who were actually being questioned last week were saying how important the wastewater programme was for them. So we published. And to some extent, we're still publishing, although it's been stripped down now, a situation report across Wales every week through Welsh Government. We're using normalised data because, of course, wastewater flows vary depending, well, in the UK and in many countries, you've got a combined sewage system. So in addition to the wastewater coming from domestic and industrial sources, there's rainwater, and a lot of it in Wales coming into the system, diluting it. So you've got to have some way of normalizing for that dilution effect. And we were using physical-chemical proxies such as conductivity. Well, the main ones that we use for conductivity, ammonia orthophosphate. So that we were able, and this was something the English programme couldn't do, we were able to get from the Welsh Water information actually every 15 minutes on the flow rates coming into the wastewater treatment works that we were monitoring. So we had flow data in real time, which was great. And so what we plotted was data, a national picture, and this is similar to many reports that you see in different countries, including the New Zealand report, where you get a breakdown of what's happening in each of the areas, and a historical timeline to show how the signal today is by comparison with what it was through a period, usually around about to 6 months or a year. And we could break the data down in terms of the areas, these 14 L2 catchments, which, although they, it allowed us to identify problems in certain geographical areas and therefore certain health boards, which was helpful to the health boards in planning things, particularly in that period through the end of 2021 and into 2021. So I touched on this, but just to sort of develop and say something a bit more about the challenges that we face, or that one faces doing this kind of wastewater treatment works, wastewater-based epidemiology. You've got to be aware that you're looking at wastewater treatment works of very different sizes. So I've just picked out two here, Cardiff Bay, and this is Cardiff. This is where the university is. And the catchment, as you can see, it's got these fingers. These are the South Wales valleys that used to produce loads of coal, had major communities in the valleys. Pontypridd is out here. This is a massive catchment coming in, all funneling in down to one wastewater treatment works down here in Cardiff Bay, which is a massive wastewater treatment works. But obviously, the signal that you're getting there is compromised in various ways. You know, how long does it take a signal from out in the reaches of Pontypridd in the valleys and the Ebor Vale, top of the Ebor Vale catchment? How long does it take for the signal to come down to Cardiff? What we really wanted to do was to break down and still want to do, and I think we'll be moving forward with this with Welsh Water, is to be able to get into the catchment and do some sampling within the catchment, as well as being able to sample at the wastewater treatment works. Kinmel Bay, so some ways they cover different geographical areas, and you've got a mobile population in Cardiff, but here you're talking about around about 900,000 people contributing to this signal. So if you've got, I don't know, say 50,000 students coming in periodically, that's a big change, but it's not huge by comparison with some places. Kinmel Bay, so this is Rhyl up in North Wales, which is popular with tourists from Liverpool and the northeast. Well, you know, Kinmel Bay can go from a relatively small population, a few thousand, to many thousands during tourist season, and a very different sort of, how do you account for that? So the effects of migration, commuting to workplace and tourism are really particularly important. I've mentioned dilution of the signal by rainfall that's running off into the combined sewer systems. We investigated things like transition time, temperature and chemicals on the viral stability, on the RNA degradation, COVID-19, SARS-CoV-2 degradation. And these were important things to do early on. And we were trying to do this kind of whilst we were developing a monitoring system to report to Welsh Government. So, and we never really got a well-established subsampling of these larger catchments, particularly Cardiff, Wrexham, Nash, Newport. But we were able to identify how we might do it. And in fact, we did apply it in one smaller catchment, which I'll come on to. So this is, just to orientate yourselves, this is South Wales. It's Cardiff down here, Newport here, which are the two, and Swansea here, which are the biggest cities in Wales. And this area here is called Merthyr Tydfil up at the top. Merthyr Tydfil is a rather steep valley. The water treatment works for Merthyr Tydfil is called Kilfinnith, and this is a larger map of the area. So Kilfinnith, the water treatment works, is down here, and the flows, as it were, the wastewater flows from Merthyr in the top, quite a higher elevation, moving down through Aberfan and then into Kilfinnith. And the colours of these areas represent the level of the relative deprivation of the communities in those in that catchment, which was an important consideration as well. So we were able to set this up and identify with the army actually, identify where the sampling points were going to be within the catchment, and then to implement that. I think we had about a week to get going on this. So we got a lot of help with Welsh Water as well to get into the network and to set up these 7 sampling points, including one sampling point that was collecting wastewater from the Prince Charles Hospital, which is the biggest in that area. And that allowed us to get information from the community, which wasn't being collected otherwise, because members of the community weren't necessarily coming forward to be tested in the eventually they called it community testing. This was towards the end of 2020. But unfortunately, as you can see from the BBC News article here, it got called at the beginning of the process, and there was 1 or 2 places, I think Liverpool, somewhere in the Midlands, somewhere in the southeast, were chosen, for this community testing. But they were told that they were going to go through mass testing, which you can imagine how the community might react to being told, well, we decided to pick on your community for some mass testing. It really made them want to volunteer to come along and give a sample. Not. So wastewater sampling was important in linking the results that came from the community testing with a signal that was relatively unbiased. So I just want to look at one aspect of the development work that we did, which was to sequence and look at variants in the wastewater, and that was a challenge. And I remember that towards the end of 21, we were told we were going to have to do this at speed because there was another variant coming in, which turned out to be Omicron. And you probably are aware that there are various ways in which the SARS-CoV-2 virus can mutate, which are indicated here: replication errors or host-mediated RNA editing, recombination. Of course, it wasn't known to be recombinogenic, and it isn't particularly recombinogenic virus, but their recombinants did occur, and these created variants which were referred to as VAMCs or VOCs. VAMCs, variants of and mutations of concern, or VOCs, variants of concern. And could that be monitored effectively using PCR approaches? Well, to some extent it could, and it was being. So remember, way back we started, the first variant to appear after the Wuhan was Alpha, and then Beta, and then briefly Gamma, and then Delta, and then of course Omicron. I'll come on to that. All changing the spike protein and the specificity of the spike protein, giving concerns about how transmission and infection might be affected by those mutations. And this panel here is just an indication of the huge sequencing effort that was going on across the world. This is a GISAID compilation of data from a whole host of countries. And there was an effort in the UK that was called the COG UK, brought together by Sharon Peacock, very quickly set up to do sequencing of clinical samples. But we were asked, can wastewater tell us anything? So we looked at qPCR, and of course you can create relatively easily assays for the different variants once you know something about them. But there were problems with Omicron and its sublineages, and things were changing so quickly that you had to be changing the assay almost, as soon as you got an assay that was tested and in place, you were trying to think of a new one. And the problem with the PCR approach was that you were looking at very often, sometimes SNP artifacts and polymerase errors, false positives, in other words, that had to be carefully controlled. We have published relatively recently work that was done by one of the postdocs on the project, Xin Sheng, who developed what was called a Varlock assay based on loop-mediated amplification, LAMP, rather than PCR, and was more sensitive, using a sort of CRISPR-Cas sequence recognition system that was more sensitive. But really, the push was on to SNP assays were useful, and the push, but limited, and the push was really on to do genomics to provide a gold standard. And so we managed to to do that. Many other groups across the world are able to do it with their own methodologies as well. And we started to implement it towards the end of in reports going to Welsh Government right at the end of 2021, which was good. It was timely, really, because there we had Delta throughout, and then suddenly over everything happened over holidays, bank holidays, Christmas, Easter. You knew something was going to happen over that time, and sure enough, over Christmas in the UK, 2021 to 2022, Omicron came in, and it really started to change quite quickly. So you had this period when you had single lineages, a lineage would sweep through and then it would dominate the picture as far as the wastewater was concerned, and that would be reflected also in the clinical sequencing. I'll show a comparison in a moment. And for a period, you had BA1, Omicron BA1, the World Health Organization stopped giving them these Greek letters and switched to just a very confusing nomenclature, so I won't go through that. But basically, the colours tell you when you've got a variant of concern that's coming in, BA1 to BA2, and then to 3, 4. And then there was a period in end of 2022 into 2023, particularly around here, and we had problems. We had problems around about early January, which was that gives me the sweats thinking about that. But we got back on again. But this was what was called by a number of investigating teams Omicron soup. So you had so many variants in there. It was just how did they coexist in the community, and which variant was going to win out next, and it always seemed to be. It always seemed to be quite diverse. So we were happy with that. And we were using a package, and many other groups do as well, which was developed at Scripps in California called Freya, to take bear in mind, you're looking at small fragments of RNA in wastewater that's being sequenced, so copied into RT, reverse transcriptase into DNA, and then short reads that are then being compiled to produce it's a sort of a slated mechanism to produce the viral identification. And always the question in my mind, because we weren't able to get hold of the clinical data because of the limitations and there were some ethical issues as well, to compare it with the genomic data that was coming out of COG. But we did eventually, and I'll show you the comparison. So we did a lot of stats on these data to try and work out how we could model this process of these sweeps of mutations that were coming through and reflected in the wastewater. And this is just one early modeling that came out of work that one of the postdocs, Will Perry, did, where you could identify two, using multidimensional scaling, you could identify two clusters, which are imaginatively named 1 and 2 here. So 1 is the circles and 2 is the triangles. And the colors here indicate the point at which those variants, those clusters are evident in the data. So this is kind of a statistic to represent the data. And what it pointed towards was that we had statistical evidence from the wastewater of a change from this, as it were, orderly succession of variants through to about September, October 2022, to a more chaotic Omicron soup situation, which was represented by cluster 2. So cluster 1 represents an orderly succession of viral genomes, and cluster 2 represents the more random, as it were, or the higher number of variants. When we were able to get hold of the clinical data from COG UK, specifically for Wales, and you put it against the wastewater data, it doesn't match exactly, but you can just see from the colours that there is a pretty good link between the two. And we're analysing these data at the moment now. In fact, we're just about to submit a paper to indicate the extent to which, for the different variants, whether we were seeing them in the wastewater before they were being seen clinically, or at the same time, or in some cases afterwards. And it's mutation dependent. So there's not one answer, even for SARS-CoV-2. And the situation now at the moment, so this is bang up to date. This is from March. That in the UK, it's all gone back to one single variant called JM.1, and it's the same in New Zealand. Everything, and in the US, everything has settled down, and unless something emerges, that's where we're going to be. But the value of wastewater is indicated, I think, here. This is the clinical data, as it were, the COG data. And this is a log scale of the number of tests that are being carried out over time. And the red line, or the dotted red line that I've put in there, indicates 100 tests across Wales. So bearing in mind it's log scale, in the early parts of 2022, thousands or even hundreds of thousands of tests were being done a week. And that was maintained, around about the thousands, hundreds to thousands through most of the period into 2023. But now it's kind of dropped off a cliff. And some weeks we're only getting 10 samples for clinical testing a week. So wastewater provides a sort of reassurance to government anyway as to what is happening across the whole of the country. And this statistic, again, a multidimensional scaling statistic, just shows you how the patterns of the wastewater data set, even when it's not normalized for population, and then the population normalized data set here, and the COG data set, which is the variant sequencing from patients and individuals in public health surveys, how they follow each other. And I think that's really pleasing from our point of view, and helps to provide some evidence to suggest that wastewater is providing useful information, has provided useful information. I'll move through this quite quickly, but I think it's going to be important for the future development. What we want to do as well is to be able to use wastewater, as it were, to say when a new variant is coming in. Can we do that without having, as it were, priors? And can the signal be interpreted? So this was a multidisciplinary project, and on the informatics side, on the math side, Owen Jones and Arthur Morris are working on a synthetic data, or have worked on a synthetic data set, which has been generated to look like the phases that we were seeing in the real data. So a phase one where you're getting a flux, these stable lineages taking over, phase two where you've got Omicron soup, and phase three now where it's gone back to how it was in phase one, transition back to single dominant lineage states. And Arthur has taken this with a tool called ART to generate a model for the genetic drift, or the genetic changes that are happening in that data set, in that synthetic data set based on real clinical data. And you can see it's represented here in this three-dimensional space, although he's working with many more dimensions than three, but I think it simplifies things. And I'll try and walk you through this diagram in particular. So what we're seeing here is the state, as it were, the mutational state of the wastewater with time. So the colors represent time here on a time scale from 0 to 100 days, arbitrarily in this model. And the three phases that I mentioned earlier are represented here, and they're also represented along this vertical time scale. And the three dimensions here show you what's happening in terms of the mutational state of the metagenomic sample that's been taken. And you can trace a path through these three dimensions from the purple through the green yellow through to the yellow. The endpoint is after 100 days. What does that tell you? Well, what you've got is cluster formation in certain places. Here, for example, here around the Omicron soup, very briefly, and then and here. And those are points of stabilization. And when you've got movement away from those stable clusters, you've got a new mutation coming in, or an emerging mutation, an emerging lineage. So drifts, as it were, from the clusters in this analysis to an emergence of new mutations are indicated or are identified in this sort of way. And this statistic is now being used to create a Bayesian model to predict when you've got a change. So if we're starting at day 100 now, and there was a change away from that, can we see that as a new mutation? And are there without having any prior information as to the mutations, the type of mutation that's happening, as was the case with the Freya 2? We're doing for time. Okay, so I shall wrap up reasonably quickly now. We're using this for wastewater analysis for looking at antimicrobial resistance as well, which I'm particularly pleased about, because that, I think, is as much, if not more, of a threat than potential future pandemics. And so the 47 sites and 9 hospitals, the shading of the areas here now represents the 7 health boards across Wales. So we're evaluating the abundance and diversity of antimicrobial resistance genes with huge metagenomic sequencing of samples across Wales over about a period of a year now. We've collected samples and trying to look at the stability or otherwise of these signals in time and space. We've used 2 approaches actually to look at antimicrobial resistance gene markers: metagenomics, which I think is probably more valuable, but high throughput qPCR as well, where we looked at 175 markers, 175 primer sets, and we've got a huge biobank of waste samples. So if anyone wants samples, just write and let me know. We need to monetize this stuff. And we're trying to map antimicrobial resistance genes across Wales. Bear in mind, this is in the wastewater itself, the wastewater signal, and the hospital signals, which represent communities. So the good thing about this sort of data is that you don't face the ethical problems that you might face if you were going to sample individuals or use clinical data. It's also got environmental impact because of the combined sewer system. We have combined sewer overflows. So when there are storms and when the sewage system can't cope, it goes out into the rivers through outfalls, or it goes out into the estuary through weirs. And so there is a lot of public concern about this, and has been since COVID. I don't know whether the same is true. You've probably got better controls in New Zealand than we have, but it's really a live issue in the States, in Wales and in the UK. This map here from the Rivers Trust, all of these suitably brown blobs are where there is a CSO going into your river. So when you swim, you to some extent you might be swimming in diluted poo. I'm exaggerating, of course. And so we've used the samples, we're sampling rivers now and the wastewater samples. I won't run through it, but this is the workflow for the AMR analysis. And we've developed pipelines based on a program called AMR++, to identify the antimicrobial resistance gene markers, and also the microbiome or the microbial communities using Kraken and Bracken databases. So we've got 2 outputs, 2 data stream outputs, the resistome really and the microbiome, and a lot of effort needs to go into analysing that. It's a huge, huge data set. And it impacts on One Health. So, traditionally, until the last 10 years, I suppose, we've focused public health is focused on the human experience and human communities. But it's much better recognized now that if you just focus here, then you're only seeing a partial picture, because of course there is evolution of microbes in the environment, in animals, and whether they're wild animals or livestock, that is going to have an impact on human health, and vice versa. And so unless you're considering all of these compartments, you're not going to get a full picture as to what is happening, for example, with antimicrobial resistance. And, well, this graphic, which is taken from Larson and Flack review relatively recently, just illustrates the fact for most antibiotics, you're in polluted environments, you're looking at concentrations of actual antibiotics in those environments, in industrial waste, in untreated hospital effluent, and to some extent in untreated municipal sewage. You're looking at concentrations of antibiotics that are actually present in those samples, which are at or above the typical minimum inhibitory concentrations of those antibiotics for the organisms that they're meant to kill and use for treatment. And rivers and seas, although they're below those MICs, they're nevertheless selecting for antimicrobial resistance in the natural environment and the engineered environment. One of the data that we'll be publishing shortly has been submitted for publication. This is just one figure from Reshma's paper that looks at antimicrobial resistance marker abundance and antimicrobial resistance marker diversity in hospital samples, and then the wastewater inflow or inlet associated with the hospital, and then the wastewater outlet, which is going into a river. And although abundance, and this is a linear scale, although abundance and the diversity represented by the Shannon index are reduced as a result of going through the wastewater treatment works and at the outlet, you're still getting substantial markers, antimicrobial resistance markers. You're still getting AMR going out into the rivers, which will have an impact. So this just sort of, so this is the political side as you don't really want it. And there's an election going on in Wales. But before that, last year, the health minister, Eled Morgan, who was really very supportive and very helpful, in May 2023, opened up the new lab in Bangor, or it's a refurbished lab. So there's a lot of fanfare associated with that. And then a few weeks later, there was a financial crisis in the UK because the Chancellor decided that the government's going to pay the public sector pay award 6%, and that money had to be found within the year. And Welsh Government was thrown into crisis. And so on the 31st of July, which isn't that long after May, we were told we had to shut down completely, that we were given a month to close the project. And then, and Eled Morgan was really upset about this, but felt that there was no choice. And then a new variant seemed to be coming in, and was coming in fact, into, and there in the UK, new COVID variant, new SARS-CoV-2 variant, and she agreed under, she pressurized her colleague cabinets, I think, to agree that there should be a resumption of the wastewater monitoring in Wales, but what was called at a lower scale. So we had to devise a plan to shift from about 50 sites to something like 11 sites. But the data that we had was excellent in identifying which were the sites to concentrate on. And then just last week, Eled Morgan was giving evidence at the COVID inquiry, and as I said earlier, she was saying how important that programme has been in assisting in the policy decisions that were being made by Welsh Government. But we're running until the end of March. We don't know what's happening end of next week. It will go through another hiatus. I think it's been the First Minister for Wales has been pretty well elected now, so it's likely to be somebody who has a science background, and that's great for us. But we honestly don't know where we're going to be going with in terms of the programme as far as Welsh Government is concerned beyond the end of this month. And I think it's fair to say that this has been successful, but the science and methodology really from here needs more development, needs to become more robust. We got within the last 6 months, we got to a place where we had trust and cooperation between ourselves and Public Health Wales, a real partnership. And that it's a shame that we're going to have to work hard to try and build on that in the absence of Welsh Government funding, at least for a few months. But I think that's in a good place now, whereas it wasn't a year ago. But we still need to secure funding to make this what I think is it needs to be a program that is embedded and is fit for the future and has security to allow it to operate for pandemic preparedness. So we're working closely with the other nations across the UK, England, Scotland, and Northern Ireland, to try and develop standardised approaches for the future. And a UK RI grant has gone in for that. It'll only provide a small amount. And also, one of the great things to come out of this is a centre for doctoral training, which involves Cardiff, Bangor, Bath University, Exeter University, the Centre for Ecology and Hydrology. And I mention it here because it's also attractive. We can take on students from overseas, and so if anyone has an interest in this, or you know anyone who might have an interest in this sort of research, please pass the information along. And I'll just finish with a cast of thousands, and maybe I should point out, the colleagues Isabel Durance, Pete Keel, Owen Jones, and Tom Connor in Public Health Wales, particularly important. Well, Tom has got a joint appointment with Cardiff, particularly important, and Davy, of course, Davy's team, Kata and Rachel Williams, been hugely just brilliant postdocs. Will Perry, Amy Baldwin, Arthur in particular, and Alvaro on the sequencing side, developing the sequencing. The drivers, unsung heroes. And then all of these alumni. And Gareth Cross and Steve Cobley in Welsh Government, Tony Harrington, Ian Trick in Welsh Water, really, brought a team together and a team spirit, and it's been a fantastic experience. Sorry, I've gone on for far too long, but I'd be happy to answer any questions.
Very much, Jen. Question, anyone, Andrew?
Ian.
You alluded to the fact that when you looked at the wastewater testing alongside the clinical data, that there were times when the wastewater testing preceded the prediction of different variants, but there was other times when it lagged.
Yes.
Do you know why that?
No. I mean, we'd have to, but it's that's a statistic really, it's only very slight. So I'm thinking, it's not by any much more than a week or two either way, and most times it's pretty well they were being reported at the same time. Bearing in mind as well that for both data sets, you're looking at data that's collected over a period of a week, and then it's compiled. But whether there's a biological explanation, I think I'd look for something in the way in which the data are being processed rather than biological explanation. And it's disappointing because I think there was a lot of hope that wastewater testing would give you that edge.
Right.
And it would do if you have no, I mean, bearing in mind the huge amount of testing that was going on for a lot of the period of the pandemic. If you imagine that wasn't happening, of course, yeah. And if this tool that Arthur and Owen have developed could be applied to, you know, metagenomic analysis of some other pathogen, some other virus, you know, if you're looking at flu or something like that, I think that could be really powerful and important. Yeah. Josh.
So I noticed that you had the 3 phases. Is it likely, or is it likely for a phase 2 like thing to become sort of cyclical? So it goes through a phase 1 where it's the succession, and then a phase 2 where there's.
Oh, I don't think. Yeah, it could. So even now, I don't think we know. Bearing in mind, okay, I don't know. We don't know the answer to that. I don't know the answer to that. That's for sure. Whether that could happen, I think possibly it could. What Arthur is doing at the moment is just focusing on mutations in the spike protein, which after all is really for SARS-CoV-2 anyway, is the mutations in that seem to be the most important in terms of its transmission and its infectivity and whether it was competitive and took off in the population. So he might be able to, you know, to model different outcomes. But all we're looking at is what we saw actually in the pandemic, and looking at it otherwise is anything, you know. And the question is how stable is it now? Is a mutation going to come with JN1? I think the sorts of mutations that really make a difference, and there are some variants in the wastewater signal which point towards this, are the recombinants, and that can change things really, really quite drastically and quickly. But SARS-CoV-2 isn't so recombinant. It's not like flu, for example. It's not so recombinogenic.
They're interested in using this to aid vaccine development. That's sort of something that we're doing.
Yeah. Well, not the
SARS.
Yeah. And there's a group working on that. Well, many groups working. Yeah. But I can't tell you. It's not my area, so I can't really tell you much about that at all, unfortunately.
Lovely. Well, thank you, Dave.